The title of this presentation is Thermodynamic Control, Studying Characteristics of Processes from Synthetic Biology to Mathematical Physics with Statistical Mechanics. To begin with a presentation overview, in this part of the series, we'll begin by drawing the viewer's attention to some other less rigorous types of connections of statistical mechanics with other areas that I've previously done research on, and how these can relate to the notions that we developed amongst more rigorously amongst several other parts that were previously given in the series. And despite the fact that we're describing some things less rigorously in some of the connections that will appear in this presentation, drawing this equivalence between experiments and different types of thermodynamic behaviors that we would expect to occur on an ensemble level behavior for cells and other types of biological networks are very helpful for providing some type of heuristic description of different models of statistical mechanics, which can then, in many instances, be made rigorous later, given some additional details and points of discussion that we have to consider and have in mind. So we'll be describing connections between thermodynamics and genomics workflows that have connections with synthetic biology. And we'll also draw attention to experimental aspects that synthetic biologists and related areas take into account through design of various assays. And in particular, I'll provide some information for some experimental data that I had talked about that was collected from a colleague in the Lambert lab when it was open. And in the synthetic biology lab, we were interested in looking at different types of ways to make connections between forms of computation for how long it would take certain enzymes to inspect DNA or RNA targets, and as well from this collection of favorable genomic targets, how having this in mind would lend itself to statistical mechanics and thermodynamics, because the enzyme, and in particular the CRISPR-Cas enzyme that we're looking at experimental data from in these talks and in this type of analysis, is related to the fact that it has to operate within a very favorable temperature range so that it can perform its main functions of inspecting the target and pursuing a reconfiguration phase after it examines the target and determines whether it can make a certain precise edit to a gene or not. And so we'll also be discussing theoretical implications of previous works which have this type of connection with ensemble behaviors of cells grown in labs across the world. So many labs are interested in looking at how to make use of different assays, whether it be trying to measure or quantify the outputs of some biological process. And they may not know all the details of this biological process, but they're trying to make some assumptions on what would be some for sure byproducts or biomarkers that would be left over from this process, and how they can develop an assay in an experimental workflow that would help them study this phenomena further. But still, from my perspective, even as a more theoretical person, we can still make contact with these areas by describing as a sequence of steps how different processes that occur in synthetic biology are related, and ultimately they have a very strong connection with statistical mechanics and thermodynamics, and they relate to different types of characteristics and themes that we brought up earlier from a more theoretical perspective throughout the series. And so we'll conclude with remarks towards other areas of interest in the remaining presentations of this series, in which for the remaining parts of this series, we'll be interested in talking about how we can make some more type of general connections, and how for a rigorous type of argument, there would still be some types of gaps in knowledge that we would need to be able to fill in so that we can have an idea about what's going on in this thermodynamic phenomena in different phases of possible behavior, like how we study in the phase diagrams for statistical mechanics. So I'm going to provide an overview of some of the slides that I presented in a previous conference, in a previous actually two conferences, and how it relates to how this model, which is rooted in some explicit solution to a differential equation, can be examined further to determine if there's some type of more experimental realization of the data and data points that we're able to gather from solutions to this differential equation, and how ultimately it can also be shown to have connections with some more theoretical areas that one might not expect. So this is given by the fact that we're looking at a differential equation, which is a first passage time model. And as we we're saying in the first bullet point, we want to look at the exit or passage time distributions tau of x. So we're thinking about if we have a CRISPR-Cas enzyme or other types of enzymes which can inspect either some type of molecular environment or be looking to target certain portions of an RNA or DNA molecule. And from this inspection phase, there exists many types of complexities for people in the genomics areas and other related computational biology areas, because there's a certain very complicated three-dimensional structure of any genome that we're interested in looking at. And this structure determines, contradictory to fact, that there can actually occur interactions dependent upon the folding pattern of the genome between areas of the genome which would appear in terms of the base numbers very far away from each other. So we want to try to be able to gain some local type of interpretation or some local type of picture of how these enzymes would interact in different types of molecular environments, and what would be the time it would take for them to explore the base pairs or other types of molecular or macroscopic characteristics of the environment, which would be related to the passage time or passage time distribution. And this is given by the fact that it's a second order differential equation. But in comparison to second order differential equations that have constant coefficients amongst or in front all of the terms of the differential equation, in this second differential order equation, which relates to some other families of PDEs or higher order PDEs, which describe more complicated inspection processes, this second order ODE has a non-constant coefficient in front of the first derivative tau with respect to x, which is the position of inspection. Namely that we're taking the derivative of some landscape potential term and normalizing by the Boltzmann constant times the temperature. And then the remaining term is just -1 times the reciprocal of the Boltzmann constant times the temperature. And despite the fact that we have this derivative of a landscape potential term, which already makes the solution and solving this differential equation a lot more difficult, because we don't have all those assumptions on the solutions to first and second order differential equations if any of the coefficients in front of the derivatives of the solution are non-constant. So, but we're also subjecting the solution of the second ODE to the fact that we want tau of n is equal to 0, because when the inspection process over n base pairs has occurred, we want the exit time at the base pair to be 0 to signify that the whole inspection process of the CRISPR-Cas enzyme or of other type of macromolecules has finished, as well as given by the fact that at the beginning of the time inspection, tau prime is 0, 0, meaning the rate of change of the solution for the passage time distribution at 0, 0, and we're fixing n to be some integer. So we're looking at some starting point at 0, and we're evolving the boundary of the domain forwards. We're propagating forwards along the positive direction of the real line, and we're determining how the solutions to the inspection process would react, or how they would behave. And so when we have this type of ODE under different assumptions on the landscape potential term, the script u of prime of x term, which appears in front of the first derivative, we can have different types of expressions or approximations, which are pretty much the same as the explicit solutions that we would be able to get from looking at the solution to this second order differential equation with the non-constant coefficient in front of the first derivative of the solution. And roughly speaking, this approximation takes the form in that we have an absorbing boundary term, an exploration position term, and an error function double integral, where the error function f sub h sub I of u is given by an exponential function. And in the exponential function, the first part has a square root of pi times sigma squared, the variance. So it's relating to the variance or the width of the peak in the landscape that the enzyme has to exert some type of thermodynamic energy for, which is relating to another type of different type of connection and theme in this presentation of the series that we're looking at in thermodynamic control, because we're looking at ways to tune the thermodynamics favorably enough so that this enzyme can have the possibility to explore hundreds of thousands or maybe even hundreds of thousands of base pairs of a target for determining which genes it can effectively target, given the guide RNA and the PAM inspection sequence that it also has to examine before looking for some target. So this is why it's very much relating to even some other topics in computational biology and statistical physics outside of the series that I've given in other presentations, in that you can look at binding affinities or thermodynamic interpretations of binding affinities, which represent interactions between ligands or other ligands in genomes or other ligands in more very isolated small parts of the genome, in determining whether there be any type of favorable conformation that would result if we pair together different targets. And this is very much relating because even though maybe an experimental synthetic biologist, they wouldn't very so much as much be concerned with the percolation or the integrability aspects of these models, it can still be of interest because it's demonstrating how this type of more theoretical results, they still exhibit a very strong dependency on tuning the parameters very precisely, just like how an experiment wants to tune the parameters of a benchtop experiment precisely for observing different types of conformations that they would expect to occur with high probability. And the computational workflow from this type of second order differential equations, so that we can further develop on both more applied and theoretical perspectives later in this part of this presentation of the series, is that given that we have this second order differential equation in tau, we can look at admissible landscapes U, where there can be different types of landscapes in which there can be some symmetry, namely there's like an energy well amongst some base pairs of the sequence that the CRISPR-Cas enzyme or other types of enzymes have to inspect. Or there could be some type of asymmetry in which, say, between base pairs 16 and 20 of a sequence, there can be a much higher thermodynamic cost for the enzyme to unpack or to examine these different base pairs of the sequence for determining if there's high enough complementarity and high enough homology for the inspection process to complete. And so this was these types of interpretations and assumptions that we put on the admissible landscapes U, as well as our ability to assemble first passage solutions from this differential equation under different assumptions on the non-constant differential or non-constant coefficient, which appears in front of the first derivative of the passage time and the second non-constant ODE, allows us to plot different types of solutions in which we study the relation between the exit time and the base pair of the target sequence. And as we had mentioned, the intercept of the solution with the base pair axis indicates the predicted exit time for a Cas nucleus at an arbitrary base pair of the inspection sequence. So it's demonstrating that there's enough thermodynamic control and enough thermodynamic stability in this enzyme for it to be able to complete its inspection process, which can then have further downstream implications for the genome in terms of gene editing, and is very much exhibiting a more applied application of these thermodynamic consequences and thermodynamic thought that we had developed throughout the presentations of the series. And now I'm going to present part of a workflow, like I mentioned, from my colleague in the lab, David Speck, who's now a postdoc in environmental engineering. And his workflow was measuring dead Cas or non-catalytically active Cas nucleus fitness change after binding. So like how in other presentations on the channel, I've drawn the viewer's attention to the ability of binding or interaction events between macromolecules can have implications for either perturbing the genome or looking at different types of potential degradation effects that can occur to the genome. In this case, we can measure dCas fitness and draw the viewer's attention to experimental types of questions that are still unknown, which could also be interested in map to more theoretically inclined systems in determining how this type of requirement for thermodynamic stability, which appears in a biological system, could be translated into more of a theoretical area, which is also relating to the topic of the remaining presentations in this series and talking about more open questions and more superficial connections and commonalities between different models of statistical mechanics. And so the objective of David's workflow was that he wanted to determine whether the presence of new genetic material can be amplified after a virus propagates its mutations to the host genome after infection. And so from his thought, we can start by choosing some viral genome of interest, whether it be SARS-CoV-2, influenza, or Zika. And after performing some genome mining, we can enumerate a combinatorial target gene space from which we can measure the nucleus dead or catalytically inactive decast nucleus binding affinity through next generation sequencing. And in the second part of his workflow, we can identify editing target sites for the dead cast nucleases depending on PAM proximal or PAM distal placements. So he's able to look at several different types of Cas variants which each have their own type of characteristic thermodynamic control and own type of characteristic thermodynamic constraints, like Streptococcus pyogenes Cas9 or AS Cas12a or ST Cas9, as well as other variants of Cas9 and Cas-X. And he's able to generate hundreds of thousands of testable constructs from approximately 400,000 constructs. And this is helpful because we can make use of this class of enzymes and look at the first passage of monotonicity with respect to the length of the absorbing boundary, which is to say that the solution to this second order non-constant differential equation that we introduced for approximating the duration of the passage time given some length of an absorbing boundary is monotonic with respect to an increasing boundary. So it's just to say that when we increase the length of the boundary, it will take the enzyme longer, and it'll also make it harder for us to exert some type of thermodynamic control or have some hope that it could thermodynamically sustain itself throughout the duration of the inspection process. And it's very much relating to how fragile different types of conditions for integrability, as well as different types of classes of conditions that we would expect to hold for other models of statistical mechanics, how they may just hold at the critical point of the model or within a very small neighborhood of the critical point of the model, or other parameters like the temperature, the Hamiltonian that's introduced, and so on and so forth, and how that very much manifests itself in different areas of experiments that one likes to perform through assay development in synthetic biology. And this is to say that we can run the approximation with the error function because, like we had said, the solution for the second non-constant differential equation, it's given by the fact that it has an absorbing boundary term, an exploration term, and the double error integral term. And we can run the double error integral term countably many times to generate some type of approximation of the total duration of time that it would take for the enzyme to examine all of the base pairs. And with this first passage approximation, we can look at examining Cas9, Cas12a, Cas13a, and Cas13b enzymes, to name a few, from which we can look to form nucleus-specific binding characterizations of binding specificity and editing efficiency, because when we have CRISPR-Cas enzymes or other types of enzymes in synthetic biology, we're very much interested in making connections about if we have some PAM sequence which precedes some gene of interest that we want to target, how efficient would the CRISPR-Cas enzyme or other classes of enzymes be in being able to perform very targeted surgical edits? And this is to say that from the solution approximation, which we generated from the error function that's given from the explicit solution under some class of landscape potentials that appear in front of the non-constant coefficient in front of the first derivative of the solution, we can tune the parameters properly so that we can have that the duration of the first passage times over a subsequence of the middle 8 base pairs inspected is on the order of 200 milliseconds, which coincides approximately with the complete time scale in which the FN-Cas12a complex forms after FN-Cas12a examines different genes of interest, which in this experimental data from the lab was taken from E. coli. And we can fashion additional analytical relations in which we can look at higher dimensional surfaces, and from these surfaces, we can parameterize them in the height h sub I, which is given of the landscape potential, which is related to the thermodynamic cost and the thermodynamic penalty that the enzyme has to subsume or undergo when it's examining this part of the sequence, as well as the fact that there can be a temperature parameter because we're looking at statistical mechanics and synthetic biology. And between these two areas, from the synthetic biology perspective, we won't be able to have a very effective enzyme that can exert a lot of energy to be able to look at different conformations or different ways of exploring the unknown landscape. So then we have to look for ways to factor in how the components from the explicit solution come into play with either the first relation that we give here, in which we want to maximize the first passage time of some enzyme given a landscape barrier height and binding temperature, in which we look at contributions from an error function like term like what appeared in the solution, except that we change the upper bound of the integral a little bit, as well as for factors which are dependent upon contributions from both the height of the temperature. And from a second relation, from the first passage time fluctuations, we can look at how would the first passage time of two different enzymes differ depending upon other types of thermodynamic constraints that we can impose on the class of enzymes, as well as given the fact that we can look at different heights from the landscape potential and different temperatures, which will all be related to the error function like we had exhibited in the explicit solution from the second order differential equation from the first passage time background slide, as well as the fact that we have other double error integral terms that we can numerically approximate for looking at their contributions and determining how, like the order of magnitude of the fluctuation for the first passage time would be. And as a continuation of the workflow, we're able to measure RNA degradation from each combinatorial argument or target, in which we have interactions from Cas13a within a small circular piece of DNA. And within this piece of DNA, we're looking at several factors that come into play, which are related to the fact that there's a Cas13a plasmid system, and there's a repression mechanism which is related to gene expression, because depending upon the viral RNA target, there can be different types of ways in which the magnitude of the repression module within this circular piece of DNA can determine the level of gene expression which occurs later downstream in the circular piece of DNA. But if we're in the optimistic case that we have a matched sequence, then the non-specific RNA degradation occurs if we're not able to adhere to a high degree of fidelity to the sequence that we want to match, which would then unfortunately incur a fitness penalty, which would then damage the performance of this circuit in the circular piece of DNA. So this is very much demonstrating how there's a little bit of a language barrier difference when we're looking at thermodynamics in a synthetic biology perspective rather than a more theoretical perspective like the areas that we had talked about from the presentations earlier in this series. But nevertheless, we can see how looking at how different biological processes that occur in sequence corresponding to different pieces and portions of the landscape potential, as well as the temperature and the fitness penalty that we can occur at each step of the landscape potential, can be related to several different types of contributions that we want to model or more theoretically analyze from a first principles perspective. And this is very much related to another type of preprint and previous work that I alluded to in the overview slide for this presentation at the beginning, in which we were looking at an operator formalism for discretely holomorphic parafermions for the two-color Ashkin-Teller, the Lupov-1 staggered 8 vertex, odd 8 vertex, and abelian Sampao models. And these types of models have different types of connections with the types of thermodynamics that we encounter in synthetic biology, because an Ashkin-Teller model is relating to coupling two Potts models together, and it's very much used to model ferromagnetism and other types of characteristics, which are related to the same types of thermodynamics and favorable properties of enzymes that allow them to expend energy when performing different types of biological processes. And there can even be loop models, which can even relate to the types of very non-trivial and complicated interactions that we can expect to occur from complicated folding patterns if three-dimensional representations of genomes. And this is also to say that the 8 vertex and the odd 8 vertex model, as originally introduced by Pauling, who was a chemist, even have also some types of connections in how different types of properties could be experimentally investigated further, which could lead to next generations of high quality, durable materials. And this is to even say that we can even look further in that we can make a connection between all these fields and analysis of PDE, because when we're looking at different types of solutions or well-posedness conditions for solutions to non-complicated differential equations, like this third order Kamasa-Home PDE, we can run a variational quantum algorithm for being able to generate some type of heuristic behavior, like how we had described some type of heuristic behavior for systems of synthetic biology, and how it's relating to the ability of certain of RNA, it's also relating to the activity of RNA polymerase and several other types of biological mechanisms, which we can at least scratch the surface of and gain some type of behavior or some type of, general understanding of different behaviors of these systems. And so we can run the variational quantum algorithm or other types of algorithms to determine whether any classes of initial conditions exist for which the algorithm can be run in polynomial time, which can give us some type of positive information to influence the types of genetic variants that we want to test in assays and benchtop or tabletop experiments, as well as the fact that the behavior of the numerical solution, approximation to the solution, which can give us an idea as to whether the solution oscillates more or less quickly at different points of the time evolution. And this is even related to different types of thermodynamic qualities and behaviors, because depending upon the thermodynamic gradient or the range of temperatures that a certain enzyme is operating at or carrying out its natural routines and processes at, it may not be able to actually have enough energy to be able to push through with the entire inspection process or reconfiguration rate or a stage after it's trying to target some type of gene along a DNA or RNA strand or RNA portion of the molecule, which can demonstrate how some other types of more applied, less rigorous perspectives of statistical mechanics can still be of interest to rigorously further justify and examine further. And this is to say that the experimental data from my colleague David Specht in the Lambert lab was talking about various aspects of experimental readout and data acquisition, in which from the particular assays and types of combinations that he was looking at, he was looking to take different combinations of promoter barcodes or crRNA barcodes, and depending upon a control condition, which I won't exactly go into now, he's looking at whether there will be any type of fitness penalty depending upon the log base 10 of the number of counts. So the higher the log base 10, the number of counts, which is given by the boxes of this combination between the two barcodes, which is given by a lighter yellow color, would indicate that there's a much higher fitness penalty, which would exhibit some type of degradation impact, which would be related to some type of more theoretical consequence of a certain thermodynamic or a certain model from statistical mechanics not being able to satisfy or no longer being able to exist in an ordered phase of the phase diagram. And there are other Cas nucleuses from the catalytically inactive or dead nucleus Cas pool that were tested, which exhibit further unanticipated fitness effects, which is still related to some type of question which we're not still exactly sure about, which I'm sure other labs around the world would be interested, or other labs in computational chemistry or synthetic biology or enzyme design or enzyme evolution or even protein evolution would be interested, because it would help them determine what are the range or what types of configurations are stable when targeting different types of genes of interest, which would be related to, again, an unexpected increase in the log 10 of the number of counts, which is given by this crosstalk plot, which is given by boxes in which we look at different combinations of promoter barcodes. So it could also be interesting to determine whether there are any types of experiments which could be analyzed from the thermodynamics perspective and the thought of statistical mechanics, and looking at how unwanted thermodynamic interactions and behaviors unfortunately negatively impact and degrade the ability of a biological system to perform some desired output or function. And when we were performing our computations for other areas like the quantum inverse scattering approach, which we had talked about early in this series, we were talking about how we were making use of the Poisson bracket. And that's even relating to this, because we can look at different types of terms and how parameterizations of interactions of terms, like we had talked about from the L operators in 2 and 3 dimensions, it can very much be related for trying to account for and study some unknown complicated three-dimensional molecular structure, and how that could still be studied from some type of different theoretical perspective in comparing the contributions of different Poisson brackets and how they relate to maybe some types of assay development or experiments that we could look into. And this is demonstrating how when we were looking at the quantum inverse scattering approach, we were able to generate this very non-trivial system first of 16 relations that was given in 2 dimensions. And that was relating to the 3-dimensional case because we were talking about how when we increase the dimensions, we have 81 relations instead of 16 relations. And this is very much relating to how difficult it demonstrates some of the inherent difficulties of being able to generalize a model to higher dimensions beyond just the single dimension of freedom that we had in this model for the first passage time for studying the diffusion times and other behaviors of macromolecules from synthetic biology. So this is just to say that this type of characteristics that we saw in statistical mechanics, in which we have a lot more complicated structure and also connections to algebra and all of these other areas through integrable probability, even if we're looking to make use of some application of statistical mechanics from more of a pure, from less of a pure and more of an applied perspective, there's still going to be several types of difficulties in trying to interact and model three-dimensional interactions with the genome or other biological entities from these CRISPR-Cas enzymes or other classes of enzymes. But reassuringly, all of the types of things that we have, as it will be demonstrated in the next presentation of the series, which is getting closer to the end of the series together, there are several types of connections between the 6-vertex model and the types of forms of the weights that are given under reflecting boundary conditions, which are another class of boundary conditions that we'll discuss in the next presentation of the series, which share many similarities in the 2-dimensional quantum inverse scattering approach. So it's just to say that even if we're looking at some more of a biological perspective, we can still look to make use of some type of 2-dimensional intuition for studying a higher dimensional interaction in 3 dimensions. And the sample space of the D22 spin chain, which is the lower rank spin chain, which we had presented on earlier in the series, which we had used to motivate different types of characteristics of the beta equation, as well as conditions that we use for the algebraic beta ansatz to generate some ensemble solutions to the beta equations that we can analyze with the root density approach, is still a very helpful type of model for also looking at interactions along a single degree of freedom, like how we had mentioned that single degree of freedom that the enzyme can propagate along when it's investigating different parts of the genome, because this spin chain with 38 possible configurations in its sample space is able to give us a pretty broad ensemble and character and possibilities for different types of conformations that enzymes or other types of biophysical systems can assume when interacting with DNA or RNA. And then finally, for 3-dimensional interactions, it could be of interest to make use of triangular ice model for making sense of some different types of biological processes and how that can be related to molecular dynamics simulations. So the concluding remarks is that we discuss how thermodynamic characteristics from statistical physics connect with ensemble behaviors of systems in synthetic biology, and we drew attention to my colleague David Speck's experiments, in which he was looking at measuring fitness penalties or fitness impacts, which are related to degradation of reverse transcription and other processes in RNA and DNA, which was consisting of the fact that he was collecting measurements from pair of barcodes on crosstalk maps and looking to color each one of the boxes of these crosstalk maps, either from one extremity being yellow or the other extremity being blue, which is demonstrating the grading of the fitness impact or the fitness penalty for that particular combination of barcodes. But on the more theoretical side, we drew the viewer's attention to how relations from complex analysis like the Cauchy-Riemann equations, which is involved in the parafermionic observable reference, hold precisely at the critical point, and other also under very other stringent assumptions on the parameters, and how perturbations at this point, much like thermodynamic perturbations like we talked about from the fitness penalty in synthetic biology, ruin order in a statistical mechanics ensemble. And that can also even be a biological type of ensemble that we're trying to look at and study through assay development or through very specific types of assays for measuring biomarkers of interest. And in the last bullet point, we alluded to topics of discussion in the remaining presentations of this series. And thanks for watching.